points are important for the interaction. Now we talked about diagnostic tests, okay? And they're quite important. Why? Because if we cannot do diagnostic tests, then you cannot find, you don't have a good diagnostics, so you cannot treat the patient. So in order to treat the patient, you need to realize what's exactly wrong with the patient. I'll give you a life scenario of my dad. He was in the hospital 2-3 days, and he didn't know, the doctors didn't know what was wrong with him, okay? And literally the test for other organs such as kidney keep getting worse and worse, but they didn't know what's wrong. They thought maybe it's cancer, okay? But then they were like, no, they thought it's melanoma because it's very invasive and it targeted multiple organs. Then they were like, no, because it's happening so quick, cancer cells, a little bit slower at least. Then a good doctor came, and he had this ability to not just follow protocol, because protocol tells you as soon as the patient is there, get the CBC and blood test and do a bacterial culture and everything. Bacterial culture, how many days it takes to come back?
3 days.
Yeah, few days takes. So until then, what the patient should happen? You don't know. Yeah, it's going to get worse and worse and worse. So the bacterial culture test was not out, but this good doctor came and he said, okay, fever, sign of infection. And he started not going necessarily through the protocol. Started thinking, applying the knowledge. The answering to your question, what should I read for the exam? You just read as I said, if you read the exam questions only, for all of your medical degree, you'll get a pass mark. Yeah, just read the exam in any university. Read the exam questions of all the. Or is this good enough? No, not at all. You just memorize some questions and that's it. You don't have no knowledge. What you need as a good doctor, you need to have knowledge, and then you have to have the next skill set to apply the knowledge in different scenarios. Because a patient comes right now is different to another patient. Yeah. So deep understanding is very, very important. Because if you understand deep knowledge, you can apply it to different things. You can have better detection and diagnostics, yes, and better treatment. Yeah, that doctor came. To be honest, he saved my dad's life because they were going to inject him with something else, and if they injected him, he would pass away. Okay, because it makes the situation worse. He came straight away, looked at it, read it, and as soon as he made his diagnostics and thing, he said, "I'm sure it's infection. I'm sure it's this." After half an hour, the bacterial culture test came, and he was right. Okay, that guy is a good doctor. Not everyone with a degree title or DR is a good doctor. Yeah.
Yeah.
Right. So even likewise with these experiments and detections, just doing the tests, or some doctors just give loads of tests and X-ray and all that. That's not necessarily a good thing. You need to use your brain, apply your knowledge. That's why deep understanding is very important. Okay, back to this. So for detection is important, and now you realize the importance of antigen-antibody interaction. Okay? In the previous lecture, so 2 lectures before, I talked about monoclonal antibodies and that we can have them as drugs. Yeah? But now we're more talking about diagnostics. Not as the one other thing that we can use antibodies for is to use them in this enzyme-linked immunosorbent assay, which is a test that we can do to calculate the concentration of antibody or an antigen, okay, or even a cytokine or a molecule inside blood. So they get the blood or even from cells, okay? And we want to know if these cells make specific antibody or make how much of it, or if they secrete a cytokine or whatever, we can use ELISA. How ELISA works, if it plays,
works. ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform...
So again, remember the plate I told you? 38 4-well plates? Here it's around 96 well plate we use, okay? And what do they do? On the bottom of it, we coat, okay, the antigen that we want, okay? And now we want to know if in the patient's blood, if we have the antibody against it or not, okay? Let's continue.
The assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells.
Yeah, all clear now. So we get the blood, okay, and we load it onto this well, which we have antigen X, and we want to know if they have antibody against the X or not. Okay, and the first step is we add the serum from the blood onto here. If they are there, they will attach, and what we'll do, we'll wash the excess antibodies. Okay. Oops.
Wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color. The development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted and the amount of antibody captured in the wells decreases. The titer is the highest dilution with definite color.
Right, so did you understand it? We get a plate, something like this, okay? We coat with it antigens, right? And then what we do, we take the blood, we take all the antibodies off from the blood, from the plasma, we coat it onto this, and we want to know if we have antibody against the antigen X or not. We have to do multiple wash steps because any excess antibody should not be there. And antigen-antibody interaction is very specific, okay? And what happens to the bottom of that antibody, okay, we have an enzyme, which that enzyme, if you give substrate to it, it changes color. And what will happen? Based on how much antibody we have, which the back of it has an enzyme, and based on the gradient of the color, we can realize which patient has higher amount of antibody. Okay? And what we can do if we make a standard concentration, we can even find the exact concentration, how much antibody is that patient has. How we need to do that? We put them in this machine. It reads the amount of absorbance, and it quantifies it. So based on that, for instance, if I know a standard which is, let's say, 10 microgram of antibody is in this sample, and I put it, and it becomes, for instance, this much. 10 microgram is this much. This is 20, 40, 50 micrograms. And let's say my unknown sample is here. Based on that, I can get an amount, and I say, okay, that well there is 5 micrograms of antibody. Yeah? So this is ELISA. And how it works, just like in the movie, you can have direct assay is when we have the antigen and we have the antibody attached to this, okay, directly to see if we have the antigen or not. Indirect assay is what happened in the movie, so it puts antibody, then secondary enzyme. Or capture assay is the opposite. We want to know if we have the right antigen or not. So what I do, I get the blood, I want to know if I have corona antigen in me or not. We get the plasma, okay, inside that soluble format, we coat it onto antibody. Instead of antigen being coated onto the plate, it's antibody. antibody against corona. If the corona antigen is there, it will get stuck. Then I have a secondary antibody against the corona. I put it there, and I can put it, just to be on the specific side, I can put a secondary antibody against that tail with the enzyme, changes color, it means I do have antigen of corona in the patient. Okay? So these are different types of tests we do. I don't think you'll do ELISA in your practical, by the way. Another test, another application of antigen-antibody interaction is for immunofluorescence. Okay? So this works similar to ELISA, but much easier. For that, what we do, you can have a glass slide, we put cells on it, or you can have a tissue, for instance.
Under a microscope?
Yes, well done. And what we do, we use antibody against CD4, or CD5, CD8, whatever, okay? But to the antibody, what is attached? Fluorescent molecule, okay? And the microscope that we have is a fluorescence microscope. It's not normal light microscope that we have, the normal light. It shines, gives a fluorescence, and it detects fluorescence. And what we can end up, we can see all these different features. For instance, in this one, how many colors we have? We have 3. We have green, we have blue, we have red. And they're all overlaid into each other. For each of them, we have to use different antibody against different things. Yeah? So antibody with a fluorescence, for instance, maybe it shows CD4. You can see the content of CD4 around the cell number. Let's say antibody against nuclear content or chromosomes, it will be shown as red fluorescence, and then you can have them all together. And this is another application of immunofluorescence. Mostly in the hospitals, they use it for cancer diagnostics, because they have different markers, and they want to know in exactly that specific biopsy if they have a specific tumor antigen or not. And they use immunofluorescence. Again, the basis of it is what? Antibody interaction with antigen. Antigen is in the tissue. Antibody, we add it as a sample. Yeah, so we have antibody against tumor antigen. Label it with purple fluorescent molecule. And if you have on your microscope, you can see purple dots, means the patient has tumor antigen. Yeah? And that's immunohistochemistry, as I mentioned. We put tissue sample or cells onto a glass slide. And for immunofluorescence, is the previous one. Histochemistry is when we have, we put the cells, and then we put the antibody to see if we have, like, let's say, tumor antigen. And then we put the secondary antibody, similar to ELISA, it will have an enzyme, and then, therefore, it changes color, and it gives us something like that. So, in these 2 slides, you can see cardiomyocyte, and the darker region, because of the substrate and enzyme we have, it shows a browny color. Okay? So, it's showing wherever is brown, and it's much more brown here, it shows complement protein. Why? Because the antibody is against complement protein, and it has that enzyme substrate, it turns the color more brown. And using normal light microscope, we can take this picture. Not fluorescence. Fluorescence is this previous one. So this is fluorescence, this is normal light microscope, but it uses the enzymes similar to ELISA. It changes color. Right, the test that you'll be doing in your practical is more this, immunoprecipitation. So one of them for immunoprecipitation is when we have soluble antigen and soluble both soluble. Yeah. Antibody usually is soluble because it's secretory, yeah. So antigen should be soluble as well, and you can see if we use the test bottom precipitate, you can see it. So it's like this, but when you centrifuge it, it will form that immune precipitation. Okay? And I think you'll do this experiment, immune precipitation. Usually, previously we used to do. Ring test is a test where we actually, what we'll do, in a test tube, first we put the antigen, we put in a solvent, then on top of it we put antibody, or vice versa. What will happen? The antigen antibody, if they're recognizing each other, they will come towards the middle and they form that immune complex. Yeah? In the middle. And you'll see like this. Okay? So loads of antigen, antibody at the bottom. First we add antibody with no shaking, of course. So just load the antibody and then gradually add antigen. And what will happen is something like this. Can you see? Yeah?
Yes.
This immune complex or zone of equivalence forms in the middle, and to this we call gray test. Okay? So the precipitation happens in the middle, and we know that the antibody and antigen interact with each other. Okay? So this is another test that they do. I don't think you do this specifically. Another test that you'll do is immunodiffusion, and it's quite interesting. So in this test, what will happen is, you remember like I said jelly, we have the jelly?
Yes.
So we make a jelly, but inside the jelly, you can either put antigen or antibody, depends what you want to do. Yes, like agar gel, okay? Inside it, in this scenario for instance, we put antibody. So all the antibody is bound onto this agar gel, okay, and stuff. We make one single hole in the middle, and we punch little holes. And then what we do, we load antigen. What will happen? The antigen start going towards the agar. Okay? And if it interacts with the antibody that we have, okay, so a patient, we can put a sort of, I don't know, plasma of a patient, and we want to, or a drop of a blood, we want to make sure if inside the blood they have the antigen against the antibody we have or not. What will happen? The antigen from the blood start migrating out, and if it actually forms an immune complex, you'll be able to see it. So this is showing the agar gel, okay? And this shows how much they have traveled from different patients, yeah? 1, 2, 3, 4, 5, 6, 7. And based on the diameter, you can find the concentration. How? If you actually use a standard. So for instance, this is a standard usually we'll do. So on the number 1, for instance, we say we loaded 100 microgram of antibody or antigen. The other one 50, the other one 30, 10, like 20. Because we know the standard and we know what size they make, we can make the graph, and based on the graph, we can measure the diameter of this, put it on the graph, and it tells us patient 6 has how much of that antigen that we want. Yeah? And this is the case. So using these rulers, you can find the diameter. Okay, it's put up there. Then you put diameter square and antigen concentration of the samples you have. And then, for instance, the diameter of this, okay, let's say, diameter square of this is here, so we can find the antigen concentration is what? This is a test, I think the first test you'll do is this. Okay. Why it's important? 1, it can tell us if a patient has antigen towards a specific antibody, or the opposite. If we put inside the gel, remember, let me go back here. What did I say? In the agar gel, what did they put here? Antibody. And in the middle, what do you drop? Blood, which we want to know if it has the antigen for that. What we can do, we can reverse it. Okay? We can put antigen instead, and then add the antibody in the middle, and see if you have antibody against antigen or not. So it depends on you, how you set up the experiment or what you want to do. And based on the diameter, as we mentioned, we do diameter square, and it can give us the antigen concentration. Why this is important? 1, it can tell us how much of an antigen or antibody a patient has, or if it has at all or not. Because if it doesn't form a ring or a precipitation like these, it means they don't have it. Yeah? But if it forms a ring, that immune complex is formed, it can tell us how much of it it has. Why, how much of it is important? Because based on that, we can detect if the patient has disease or not. So right now, for instance, if they do, you might go and do a test for antibody against, for hepatitis B. Okay, based on how much antibody you have, okay, or how much antigen you have, they realize if you're infected or not. And based on that, right treatment.
So we will
Otherwise, wrong treatment.
for the test, we will take from our blood.
They take your blood, and then they put it in these tests. They measure the antibody concentration, antigen concentration, and based on that, the doctor can diagnose.
You will give me the antigen or
Yes, you have it in the kits,
okay.
so you can commercially buy it, okay, and set up the experiment. Right. So the other test, which is quite interesting, is a little bit mathematical, more than the previous ones actually. The previous ones are okay as well. Is immunodiffusion, and one of them is Ochterlony double diffusion. And what will happen in this test is, firstly, we want to check if we have similarity between antigen or not. Remember shared epitope and all that? So this is a test we can do, okay? And what we'll do, we'll have an agar gel, and we make holes in the middle, and we can load different things at the same time. So maybe we can add antigen here, patient 1 antibody, patient 2 antibody, patient 3 antibody, patient 4, 5. We can do multiple tests all in one, okay? And so this is size of a petri dish, is like this size, okay? And this cloud is the agar. So we put antibody in the middle, and we have a control antigen from different patients, for instance. And what will happen through all of them, they started diffusing it out, okay? And if they recognize each other, they create identity band, which is like a curve type thing. If it's partial identity, I'll go into them in more detail. They create a spur thing coming out. Or if there is no identity, they create sort of an intersection. Okay, we'll go into each of them. So let's say in the middle part here, we have anti-A antibody, okay? And we have two antigens A. This anti-A, if it recognizes the antigen, antibody A, if it recognizes these two antigens, what will happen? They will diffuse out similarly, okay? And they create this line. Okay, it's like bent line, curved line. But if it does not detect them, because we have anti-AB antibody, this is antigen A, this is antigen B. Okay? It will create an intersection. It will not fully recognize it. So it will create an intersection. But if you have anti-AB antibody and it recognizes that, it creates a partially identical, or it creates a spur. We say to this part here, we say spur. Meaning it recognizes this, but it doesn't recognize that. Why they're important? In a scenario where we have multiple antigens and antibody in a patient, we don't know which one's which. We used to use Ochterlony double diffusion. So again, just to emphasize, we have, if it creates this curve, it means identity. It recognizes both of these antigens. Okay, so band of identity. Partial identity is a case where we have spur. So antibody XY recognizing this side, it creates a spur over here, but it does not recognize that as fully. If it doesn't recognize either, in a sense, it creates this intersection, so reaction of non-identity. So we know that this patient, for instance, does not have the antigen against XY. It has X and Y, but not them together. Right. Any questions? Let's do a summary. So we talked about antigen-antibody interaction. What are the key features for the binding of them? Okay. And then the 3D structure of it, molecules are important for it. We can have cross-reactivity and shared epitopes and cross-reactivity. Then we talked about What else we talked about? Oh yeah, when you have excess antibody is called what?
Access,
post.
Excess antibody.
Sorry, prosome.
Prosome. Then in the middle,
Equivalence.
equivalent, which forms immune complex. Yeah? So all these immune complexes, you just saw the pictures how it look like. Yeah? And then when you have excess antigen,
and you talked about the fluorescence.
yes. Then what else we talked about? We talked about affinity, the strength of antigen-antibody interaction with one antibody molecule. And we said antibodies, maybe they're bivalent, one antibody attaching to the same epitope twice. Monovalent is when we have fragment antigen binding. Multivalent, for instance, IgA, if it's a dimer, it can bind to 4. Polyvalent means many, is like IgM. So the affinity is between single, but avidity is between the whole molecule, and it's much more stronger than just a single one. And we did that experiment of immune complex formation here. Then we talked about the diagnostic test, that antigen-antibody interactions are very important. The first we talked about was Western blot, which is to identify if a specific protein is existent and how much of it. How we can do is get the cells or tissue, whatever, break them into protein content, put them on a gel so they will separate it. Then we load a secondary antibody against our whatever molecule we have with the enzyme, just like what you saw with the ELISA. Then we talked about ELISA, which we can use to find how much of an antibody we have in a patient. For that, we need to coat it with antigen, the wells, or the vice versa. We want to know how much patient has antigen. For that, we have to coat it with antibody. And then the secondary antibody with the enzyme and all that. Then what else we talked about? We talked about flow cytometry. Yeah? So we can phenotype different cells and say how much of each marker each cell has. Yeah? For instance, for the case of HIV, a good test would be CD4 percentage. Then we talked about, what else was there? The agglutination test. Okay, agglutination test is when we have soluble antibody and particle antigen. Yeah, solid antigens. Hemagglutination is doing agglutination tests on red blood cells. Okay, such as blood typing we're using. Then the other one, immunoprecipitation, is literally what you are seeing the pictures right now, is when we have soluble antibody and soluble antigen. And so on the test that we saw here was one was immune diffusion, that we put antibody or antigen inside a gel, and then we put the reverse of it, and we want to, based on how much diameter it is, we can find the concentration. Then the Ochterlony here, which it's definitely good for mixed, if we want to see if it has mixed, we have a mixed reaction and we want to check to see if they have identity or not. If they form a curve, it means they recognize both. If it forms a spur like this, it means it recognizes one partial identity. If it creates an intersection like this, it doesn't, no identity. Okay, so the antibody is not shared between the two. Yeah? And these will be all the majority of the tests for antigen-antibody interactions. Right, thank you. Any questions?
For the paper.
For the paper, question.
Practical.
Right, the question, right answer, whether it's A or B. Let me think of the question. When we have excess, you remember on that peak that we had? When we have excess antibody, okay?
Yes.
Is it prosome or postome? A, is it prosome or B, postome? So you remember, excess antibody, then equivalence, which forms immune complex, and then goes back down, excess antigen. Okay?
Prosome or
Prosome Opposed to. So what's the right answer? When you have excess antibody. Anyone?
It's
A.
Proso.
It's
Proso.
A.
Yeah, Proso.
My answer is always A. Yeah, it's
Is
frozen.
it right?
Just write A.
Can I ask you about the
Could you
writing exam?
explain